Okay, so guys, today we are starting the topic of gene therapy. In the last class, we've already done microarray technology and bioinformatics. And now we move on to the topic of gene therapy. With gene therapy, we will be doing, first of all, we'll be trying to understand what gene therapy is. And then after that, we will be going into the indications of gene therapy, what kind of diseases can be treated by it, the social and the ethical and the moral implications of it, so on and so forth. So let's start. So Cambridge definition of what is meant by gene therapy. So I'm going to share some Cambridge questions and their relevant answers. The Cambridge says that the gene therapy is done to alter the genotype by inserting the dominant or the normal allele, not gene. Again, remember where you have to use the word allele and where you have to use the word gene. So a therapy that alters the genotype by inserting the dominant or the normal allele, not the gene, into the affected or appropriate cells with the help of a named vector. You all know what vector is. It's a vehicle which will transmit the DNA from one organism to the other. And by the help or by the use of recombinant DNA technology. Whenever the word recombinant comes into your mind, you should imagine a plasmid or a viral vector that is made up of DNA from at least two or more than two sources. So the therapy that is meant to alter the genotype by inserting the normal or the dominant allele. अब यहाँ हम normal और dominant allele इसलिए कह रहे हैं कि most probably, let's say, if it is a disease like cystic fibrosis, which is an autosomal recessive disorder, or albinism also. So in that case, since this person is homozygous recessive, so he develops the disease. So if technically we could insert a functioning normal dominant a functioning normal or dominant allele, we could get rid of the symptoms. We are not replacing the recessive allele with the dominant allele. We are just inserting the dominant allele. Please keep that in mind. Okay? I'll show you now another question. The previous one was, what is meant by gene therapy? Now the question has been changed a little bit. Outline the goals of gene therapy. And let's say if this question was of 3 marks, I think ye 3 marks ka hi hai, right? So let's see the goals of gene therapy. The goal of gene therapy is to insert a functional, normal, dominant, correct, jo bhi aap bolo sahi hai, allele, not gene. That's why the word allele has been underlined over here. And then you are not supposed to write this down. You are not replacing, you are not removing a faulty allele or a gene. You are just inserting a functional, normal, dominant allele. Either you say functional or normal or dominant or correct, both, all, any of these options are correct. Why are you doing this? Because you want to obtain a normal polypeptide. For example, if we want to insert an allele which is clotting for factor 8, so we want that allele because it will make, or it is coding for that particular polypeptide. Why are we doing this? Because the question was outline the goals of gene therapy. We are doing this to reduce the symptoms of the disorder. We want to restore or modify or enhance cellular function. And we want to increase the quality of life. Remember in the last class, I was talking about life expectancy, life survival, the quality of life. So at least we can give for as much as how long this person lives, we can improve the quality of his life. How does gene therapy work? What is the basic outline of gene therapy? Now, since you know from the chapter gene technology, so much we have studied up till now, कि कैसे हम एक organism से gene निकालते हैं, how do we insert it into a plasmid, where do we obtain the plasmid from, and then what do we do with that recombinant plasmid, right? How are we going to isolate those bacterial cells which have taken up the plasmid, so on and so forth. So now you all know, right? As a general rule, this slide is telling you a general rule. As a general rule, most gene therapies studies, a normal gene is inserted into the genome to replace an abnormal disease causing gene. मतलब इसकी एक आपको बता रहे हैं, like the general outline. For that, for example, if I want to insert a normal allele, ठीक है, I will want to use a vector. I can't do that without a vector. So a carrier molecule, which is a vector or a vehicle, must be used to deliver this gene or this allele. In fact, I think ये जो slide है, I have taken it from Google. So I think I should remove this word gene from here so that there is no confusion. Let's remove this replacement thing. Okay, you are just inserting a normal allele. This should be the correct answer. Okay, and then over here also the allele, right? The most common vector that we use is a virus that has been genetically altered to carry the normal human DNA. Of course, what we are inserting is the normal human DNA. हम normal human allele insert कर रहे हैं so as to increase the quality of the life of the person, and for that we need a virus. Usually viruses are used. Viruses have evolved a way of encapsulating and delivering their genes to human cells in a pathogenic manner. Now, the problem with viruses is we know that there is no virus which is saprophytic or which is beneficial for the human beings. All of them are obligatory parasites, which means they are pathogenic. They produce diseases. So whenever we are using viruses as vectors in gene therapy, we always have a risk that the virus might cause number one mutations, or it might even cause a disease. And you know, viruses like even HIV virus sometimes is used. Lentiviruses are used, adenoviruses, retroviruses, they are all used. Now, although, as you can see in this, what I have written over here, that while we use viruses as vectors, we always try to treat the virus chemically in such a manner that the pathogenicity of the virus is reduced and the antigenicity is retained, meaning that this virus will behave as a virus. It should be capable of carrying that human allele, the DNA, but it is treated in such a manner that it will not cause havoc inside the body, or it won't cause disease. But this is a very difficult thing to achieve, right? We have to keep a balance between the maintenance of the antigenicity and the reduction of the pathogenicity. So we can never eliminate the 100% possibility of the fact that the virus is not going to cause any problem, right? That risk will always remain. But as you can see in the bullet points over here, that the scientists manipulate the virus genome to remove the disease causing genes and insert the therapeutic ones. Target cells, such as the patient's liver or lung cells, are infected with the viral vector. Acha. And these are the common viruses which are used. This is just a general outline. Details I'll share with you. Acha. What are vectors? It's just a revision from before. You all know that vectors are carrier molecules which are employed to enhance the gene transfer efficiency in gene therapy. You have to optimize a vector, right? One must consider the host immune response. One must consider that when he's using these viruses as vectors, he must target specific tissues for long-term gene expression and regulation of the gene after insertion as well. See, this is a very difficult thing to achieve. Okay, you have to introduce that particular allele into the person's body by using viruses as vehicles. At the same time, you have to, make sure that the viruses don't elicit, that the bodies of the human being does not react or does not reject the virus, does not produce an immune response. And that when you are introducing that allele through this viral vector, the viral vector should have a capability of targeting only the specific tissues. For example, कि अगर मैं cystic fibrosis में gene therapy दे रही हूँ, so in that case that the allele should go and target the specific organ which I want to treat. For example, the signs and symptoms of cystic fibrosis are more concentrated in the lungs. So then that allele should be inserted or should be taken inside the body in such a manner that it should go and affect your lung cells or start, some improvement in the lung symptoms. Okay? And then this is the most important thing to achieve कि आपने जब virus डाल भी दिया as a vector और वो vector carry कर रहा है वो allele, then somehow ऐसा कोई तरीका हो कि you can also regulate the gene after insertion. That's the most difficult thing to achieve, right? But to sum up, the bottom line is that both the viral and the non-viral vectors have been used though non-viral have a decreased transfer efficiency. अच्छा, now problem ये है कि If I'm using a viral vector, the efficiency of the transfer of the DNA is very high. But the risk of the immune responses, the risk of diseases caused by viruses, rejection by viruses, cancer, mutation is very high. और अगर मैं नॉन वायरल वेक्टर लेती हूँ तो उसमें ये है कि जो सक्सेस रेट है वो कम है, राइट, बट the other side effects, the unwanted side effects, their risk is minimized. So that's the problem. Okay? How does gene therapy work? Sometimes we can also use non-viral vectors. And if you remember, when I was teaching you the manufacture of insulin, genetically engineered insulin, I did take the name of liposomes. So liposomes are also vectors. In fact, I told you कि तीन तरह के vectors use होते हैं: plasmids, viruses, and liposomes. Plasmids we've done, ठीक है, and we also did study कि plasmids के use करने से कितना बड़ा risk होता है कि when we are using antibiotic resistance genes as markers, that increases the risk of spread of these antibiotic resistance genes in the bacterial population. तो उसमें ये एक side effect हो गया. Viruses को अगर हम vector के तौर पे use करते हैं, the viruses will cause disease, right? They will mutate sometimes, they will cause cancer. Liposomes can also be used, but again, liposomes are non-viral, as I told you. उसमें जो है disease की जो वो है risk है, it's the least, but the transfer efficiency is the minimum. So basically, what's a liposome? It's a sphere. It's a sphere, as you can see in the diagram over here, that is bounded by a phospholipid layer. So what do we do over here? By using liposome, we can directly introduce the therapeutic DNA into the target cells, right? It can be used only when certain tissues, and it can only be used with certain tissues and requires large amounts of DNA. So what is a liposome actually? It's nothing, but it's an artificial lipid sphere with an aqueous core. अंदर का जो इसका core है, it is aqueous, and outside part is a phospholipid bilayer, and it has the capability that it can carry the therapeutic DNA. Not only it can carry, number one, but it is capable of passing the DNA through the target cell's membrane, right? If it does so, the advantage is that it has no risk of diseases. लेकिन मसला ये है कि इसका success rate इतना ज़्यादा नहीं होता. This is a diagram that very well explains that how we can use a viral vector, how we can introduce our therapeutic DNA into the vector, and how finally then that viral vector, which contains the corrected DNA or which contains the therapeutic allele, is inserted into a cell culture, right? On the left-hand side, we begin the journey, the numbers, the sequence has been numbered over here, right? It shows you that a DNA that has been modified, उसको हमने पहले से ही modify कर लिया है, जैसा modify का मतलब जिस तरह का DNA जो allele हमें चाहिए थी, we have actually obtained that DNA and we have now inserted it into the viral vector. नजर आ रहा है आपको कि इस वायरस के अंदर ये modified DNA, it's there. Now this viral vector containing DNA is inserted into a cell culture. Cell के अंदर इसको दाखिल कर दिया, and you can see that the virus is taken in by the cell and packaged into a vesicle, right? This is your vesicle membrane. The virus makes its way to the cell nucleus. सारे cell की nucleus के अंदर ही जाएगा ना, where the vesicle breaks down. Number 5 is that, see, it's making its way into the cell nucleus. Through the nuclear pore it will enter, and now the corrected DNA is integrated with the patient's own DNA, right? And then finally the encoded protein is produced by the cell. So this is a simple logical sequence in a stepwise manner that shows you how we can use viral vectors to carry the modified DNA and finally transmit it into the person's cell. And over here, the example that has been given to you is of the adenovirus. And as I showed you in the previous slide, adenovirus, lentivirus, and retroviruses can be used as viral vectors. The risk of transmission of viral diseases, the chance of mutations, the development of immune responses, and the development of cancers is also common. There can be two kinds of gene therapy. in vivo gene therapy and ex vivo gene therapy. Ex vivo का दूसरा नाम है in vitro. In vivo means inside the body, in vitro means outside the body. तो हम क्या कर सकते हैं कि we can directly introduce the allele into the target cells of the body, which we do in case of cystic fibrosis. And then there are some diseases, like there is a disease which is known as severe combined immunodeficiency disorder. It stands for SCID. In that disease, what do? We take the cells. Okay, this disease actually affects the white blood cells. So we take those cells, the white blood cells in the person's body, we introduce the normal functioning allele into those cells, and then we, put those cells back into the body. So this means gene therapy can be given by introducing the dominant functional allele directly into the person's body cells or target tissues, or sometimes the target cells are removed from the body and then they are genetically modified. So both things or both methods can be used. Problems with gene therapy, I've already discussed with you. Gene therapy is not a lifelong cure. Don't think कि if एक दफा मैंने gene therapy दे दी तो वो gene हमेशा ही रहेगी. No, it will only start producing that polypeptide for which it is coding for a short time, because it's not a part of the person's own genome. ठीक है? So it will get destroyed. So that's why the nature of the gene therapy is that it's always short-lived. So this means the patients will have to undergo multiple rounds of gene therapy. The risk of development of immune response is also there. Problems with viral vectors, जैसे मैंने कहा कि viruses can be toxic, they can elicit an immune response, they can even elicit an inflammatory response. ठीक है? And the gene therapy cannot be used as a cure for multi-gene disorders, जैसे कि heart disease, high blood pressure, Alzheimer's disease, rheumatoid arthritis, diabetes. These are all caused by the combined effects of variations in many genes. ठीक है? तो इस तरह के जो problems and diseases like these, which are the result of combined effects of variations in many genes, cannot be cured by gene therapy. Gene therapy specifically can be given to those people जिसमें एक खास कोई dominant जो है ना वो allele absent है, और आप जो है वो उस dominant allele को insert करके and you're trying to relieve the symptoms of the patients for a short time. The ethical concerns of gene therapy, details I'll do afterwards. लेकिन थोड़ा बहुत मैं आपको बता देती हूँ कि gene therapy can be targeted on your germline cells or your somatic cells. अगर आपको याद हो, germline cell means cells which are in the gamete, ठीक है, या zygote में. If we are doing the gene therapy on a zygote or inside the gamete, this means that इसमें ethical concern इसलिए आ जाता है कि we did not take permission from the zygote. That zygote will become a human being one day, right? So should this be done to a person? Is it against human rights? There are many ethical, moral, social concerns against it. So that's why gene therapy done in the egg and the sperm cells or in the zygotes is a very controversial issue. Yes, somatic gene therapy can be done. This, for example, somatic gene therapy to adults nahi hoga na. If I am suffering from cystic fibrosis, so I will go myself to a doctor and say, please cure me for this disease, or help me alleviate my symptoms. I want to be a candidate for gene therapy. So that's why jo aapki germline gene therapy hai na, it's just like making a designer baby, and there are many religious ethical concerns for it. Details I'll do afterwards. इस पे usually ethical concerns पे ऐसे question आता है, right? And then we will move on to the gene therapy, which is a scientific perspective. Scientific perspective ये है कि gene therapy can be somatic, as I told you, or it can be germline. If it is somatic, then this means that new genes are introduced into the person's body. Usually it is done in the adults. And in germline gene therapy, the human germ line is modified, which means we are introducing those alleles into the sperm or the ovum or into the zygote, conferring heritable modifications to the offspring. However, germline gene therapy is not permitted in any country on the basis that it is unethical. Okay. Under the topic of gene therapy, we will be doing some proteins which the human body requires, and there are certain diseases in which those proteins are not made, and gene therapy is used to treat such disorders. So I will just name First of all, I'll just start the discussion with some of the very important proteins which are needed by our body. Example is one which you already did, which is insulin. Second example is human growth hormone, thyroid stimulating hormone, and factor 8. Okay? All these are different kinds of proteins. Human growth hormone is needed for growth. Thyroid stimulating hormone is that hormone which will be released by the pituitary, and it will stimulate the thyroid glands to secrete thyroxine. Factor 8 is a blood clotting factor or a protein which is made by the liver, which helps in the clotting pathway or clotting cascade. Okay? So these are just examples of the human proteins that are made by genetic engineering. मतलब हम इन proteins को genetic engineering से बना सकते हैं, just like we made insulin, right? अच्छा. Going back, we are going retrospectively and we are trying to think कि जब हमने इंसुलिन बनाई थी जेनेटिक इंजीनियरिंग से तो उसके क्या एडवांटेजेस थे, राइट? द एडवांटेजेस वर that the method used was very simple. ठीक है? Why? Because हम हमारे पास कोई ethical concerns नहीं थे, because we were using bacteria. ठीक है? हमने जो एग्जाम्पल स्टडी की वो हमने बैक्टीरिया की स्टडी की. We were using bacteria, और ये तो हमने नहीं किया yeast and cultures of mammalian cells to produce these proteins. These cells, which means these cells का मतलब बैक्टीरिया ही जब हम यूज कर रहे थे, they have very simple nutritional requirements. इनके लिए तो lactose में grow करना ही काफी है इनको, right? And large volumes of products can be produced because we can allow the bacterial colonies to replicate. You saw when we were having a discussion on genetic engineering of insulin कि हम जब उसको fermenter में भी डालते हैं finally अपने transgenic bacterium को, तो we can get those bacterial colonies. Their multiplication time is very, very rapid. So we can have it done very easily. And at the same time, the production facilities do not require much space also, because growing bacteria in a fermenter does not require that much space कि अगर हम कोई और methodology use करते. तो these are just this is just discussion, ये मैंने आपकी textbook से ही लिया है, on what are the advantages of using bacteria or organisms like yeast to make genetically engineered products like insulin, growth hormone, thyroid stimulating hormone. So these are the advantages. These are some advantages, and there are some disadvantages also. वो disadvantages अभी आएंगे. Sorry, अभी advantages ही चल रहे हैं, additional benefits. कि bacteria में there is a less chance of allergic responses, right? जैसे अगर हम viral vectors use करें, अगर हम उससे compare करें, so the chance of allergic responses or immune responses from bacteria is less. There is no risk of disease from contaminating pathogens. Ethically, religiously, morally more acceptable. So when we are using organisms like bacteria for genetically manufacturing proteins like insulin, growth hormone, thyroid stimulating hormone, it has many, many advantages. These are all the examples of advantages. But there are few problems also, right? Achha, sorry, ये भी advantages ही चल रहे हैं. I can write down. These are also advantages continued. Another advantage of using genetically manufactured product by using bacteria is that जब हम नहीं बना था genetically manufactured insulin, so we used to take insulin from horse serum or from pig serum. So yes, now there are no ethical problems also. ठीक है? Less chance of allergic reaction to horse's serum or pig serum. Less chance of getting even diseases, because अगर हमको factor 8 निकालना है तो हमें बार-बार blood निकालना पड़ेगा. So the problem of getting blood from so many people is also minimized. Disadvantages of genetically engineered proteins. वो क्या हो सकते हैं? The disadvantages, as you can see, I have highlighted it over here, that when we use bacteria to produce human proteins is that bacteria do not modify the proteins in the same way that eukaryotes do. It is much better, therefore, to use eukaryotic cells to produce human proteins. See, the normal dogma of biology or protein synthesis is DNA makes RNA, RNA make protein. हम यही चीज फॉलो करते हैं when we genetically engineer insulin with the help of bacteria. लेकिन एक दफा जब बैक्टीरिया प्रोटीन बना लेता है, it doesn't modify it. You know from your knowledge that when proteins are made, post-translational modification also takes place, which means that they are folded so that they get a secondary structure or a tertiary structure. sometimes amino acids are added or removed, or sometimes prosthetic groups are added or removed. तो ये चीज बैक्टीरिया नहीं कर सकते. Only eukaryotic cells can do. So the thought came that why don't we use eukaryotic cells to produce human proteins rather than using bacterial cells to produce human proteins. ठीक है? So let's see what kind of eukaryotic cells we can use to make human proteins. You know, there is a disease known as hemophilia, which occurs because of a deficiency of the factor 8. Factor 8 is produced normally in a normal human being by means of from the organ liver. If the person does not have a factor 8 के लिए, factor 8 को normal function करने के लिए, you know that it's a disease which is transmitted on the X chromosome. So at least a person should have one of this allele. ठीक है? अब अगर ये XY है और इसके पास ये है, तो यहाँ पे तो कोई allele होगी नहीं, right? So this means such kind of a person, or females में तो होता ही नहीं है, और इस तरह का female तो the zygote dies. So if a man, he develops hemophilia and he has inherited the small h allele, how do we cure him? By manufacturing genetically engineered factor 8, but not by bacteria, by using eukaryotic cells. Let's see how this is done. Genetically modified hamster cells. Hamster, I don't know whether, I hope you know what a hamster is. So genetically modified hamster cells are used by several companies to produce factor 8. This protein is essential for blood clotting. People who cannot make it are said to have hemophilia. The human gene for making factor 8 has been inserted into hamster kidney and ovary cells, which are then cultured in fermenters. अब ये cells eukaryotic cells हैं. ठीक है? The cells constantly produce factor 8, which is then extracted and purified before being used to treat people with hemophilia. These people need regular injections of factor 8, which before the availability of recombinant factor 8 came from donated blood. Using donated blood carries the risk of infections from, for example, from HIV. Recombinant factor 8 avoids such problem. So the example that I'm quoting right now over here is the use of a recombinant factor 8 in which the bacteria has not been used for genetic engineering. Instead, the genetically modified hamster cells are used. Okay? So the advantage of using this kind of a mechanism will be that since the hamster, ovarian or kidney cells are eukaryotic cells, so इसमें फिर eukaryotic gene expression or protein modification होगी, right? Okay, so yes, we were on this slide. This is how we can manufacture factor 8. Then there is another disease. You also need to know about the details about this disease for exams. It is known as severe combined immunodeficiency disorder, SCID, right? This disease, in this, what happens, there is a mutation of the gene, which is known as the ADA gene. And mutation of this gene results in a lack of an enzyme, which is known as adenosine deaminase. And this enzyme is required for the normal function of the T and the B cells. So agar aapke paas ye enzyme nahi hai, right, because of mutation of this gene, it will result in the inefficient function of T and B cells. And T and B cells, you know from your AS knowledge, they're required for immunity. So what will happen? A buildup of the substrate, deoxyadenosine, will start taking place, right? And this substrate is toxic to immune cells. So matlab ye hai ki this is an enzyme. The name of the enzyme is adenosine deaminase. Since it's an enzyme, it must be catalyzing some reaction which is necessary for the normal function of the T and B cells. If you don't have this enzyme, why? Because of mutation of the ADA gene, there is a buildup of a toxic substance which is known as deoxyadenosine. And that substance actually destroys the your body cells, your immune cells. And it is an autosomal recessive disorder, right? So how to treat it? How to treat it? Through genetic engineering. High yields of the enzyme adenosine deaminase, which is used to treat severe combined immunodeficiency disease, are made by a genetically modified insect larva, the cabbage looper moth caterpillar. This enzyme is administered, यानी हमने क्या किया, insect larva को genetically modify किया, so that this insect larva starts producing the enzyme. And then we can sort of, extract and purify the enzyme. And then we can use this extracted, purified, humanized enzyme and administer it to patients while they are waiting for the gene therapy or when gene therapy is not possible. अब आप लोग सोच रहे होंगे कि मिस ने डिस्कशन तो जीन थेरेपी से शुरू की और फिर मिस वापस इस पे कैसे आ गई. Actually, both these topics are taught together. We teach you that gene therapy is required for the treatment of the disorders in which, especially the autosomal recessive disorders, right? Now let's say if a person is waiting for gene therapy, is a possible candidate for gene therapy, तो उसको भी हम ये दे सकते हैं, right? ऐसी treatment दे सकते हैं. Or if the person was given gene therapy and he did not respond properly to it, he developed allergic responses or reactions to the use of viral vectors, ठीक है, या फिर gene therapy उस पे possible ही नहीं है, कोई भी reason हो सकता है. In that case, we can give them for life, we can give them genetically engineered human insulin, genetically engineered growth hormone, genetically engineered enzyme, this enzyme adenosine deaminase, or genetically engineered factor 8. ठीक है, so this is giving you an idea how we can do it. But इसमें इस description में खास बात ये है कि we are not using bacteria. Yes, sometimes even animals. अभी तो हमने बात की थी ना transgenic bacterium, and then we said कि we don't want to use transgenic bacterium. We are using genetically modified hamster cells, hamster kidney and ovary cells. Then we are using genetically modified larva of a caterpillar. We can also use genetically modified animals. कैसे? Some proteins are even produced by transgenic animals like sheep and goats. Sheep and goats are genetically modified to produce human proteins in their milk. यानी हमने उनको genetically modified किया, तो when they produce milk, they have those special human proteins. For example, there are two other proteins which might be required for people who have a deficiency of these. One of them is known as human antithrombin. Antithrombin से पता चल रहा है कि ये clot नहीं बनाता. This is also required because see, just as we have clotting factors, we have anti-clotting factors also to balance all this. So human antithrombin can be produced by genetically modifying goats. Okay, this protein can be used to stop blood clotting. Let's say if there is a person who's suffering from a disease because in which he has excessive thrombosis, okay, तो उसको हमें ये देनी पड़ेगी. Or we can also manufacture human alpha antitrypsin, which is produced by sheep. And this protein is sometimes used to treat people with emphysema, right? So we can also do that. Achha. This was about gene therapy and about the use of genetically modified eukaryotic cells so as to manufacture different kinds of proteins. Then we move on to the topic of gene technology and agriculture. Okay? जिसमें we are sort of using genetic engineering to improve the quality and the yield of crop plants and livestock in solving the demand for the food in the world. यानी कि अब हम genetic engineering की एक further extension कर रहे हैं, उसकी implementation की, उसकी usage की, कि we are using our knowledge of genetic engineering and we have two objectives in mind. I want to increase the quality of my crop plant, right? I'm an agriculturalist and I also want to increase the yield of crop plant per acre or per hectare. And also if let's say if I am running a livestock business, poultry and eggs and all farming, I also want to improve that. So for that, I can also take the help of genetic engineering to do that. And the examples that have been used in your textbook are 3. Plants में 2 example use हुई हैं, BT maize and BT cotton भी शायद है, and vitamin A enhanced rice, which is known as golden rice. And then in animals, it is GM salmon, the fish GM salmon. I'm now stopping my recording here because I want to have a discussion with you, and then we will finish this bit after during the discussion only.